Here we are today with the third part of cellular respiration, the Krebs cycle. This diagram should begin to feel familiar to you. I'm hoping that you recognize some of the topics that we've already talked about. Today we're moving into the citric acid cycle. You can see it right in the center of the screen in the middle of the area of the mitochondria. And it's also called the Krebs cycle. In the AP curriculum framework, there's a reference to the Krebs cycle. You can see it here on the screen, where it emphasizes that carbon dioxide is released from organic intermediates, that ATP is synthesized from ADP and inorganic phosphate via substrate level phosphorylation, and that electrons are captured by coenzymes. Also in this framework, it indicates that electrons are extracted in the series of Krebs cycle reactions and are carried by NADH and FADH to the electron transport chain. So we are in sort of the center of cellular respiration when we are considering Krebs cycle. We have finished glycolysis. We're going to take what came out of glycolysis, process it further in the Krebs cycle. Here's a picture that shows all of the events of the Krebs cycle as a big graphic. You'll notice the rectangle on the top is the oxidation of pyruvate, which we've already discussed, but just as a reminder, remember pyruvate has three actions that happen to it and convert it to acetyl coenzyme A. Remember that a carbon dioxide is formed, NADH is formed, and coenzyme A joins the remnant pyruvate, forming acetyl coenzyme A, which has 2 carbons left from the original 3. As we look at the Krebs cycle today, or the citric acid cycle, we'll track the fate of those carbons, but more importantly, we're going to look at what happens to the hydrogens, which aren't really even drawn in on this image of pyruvate, but they are much more important in terms of energy transfer than the carbons are, because in our process of breathing, we're going to breathe out those carbons eventually. With the hydrogens, though, we're going to use them to have some energy transformation occurring. So I'm going to show you a video clip that will go through the process of the citric acid cycle, or Krebs, and then we'll tease that apart after the video is completed.
Let's follow this pyruvic acid molecule into a mitochondrion to see where most of the energy is extracted. As the molecule enters the mitochondrion, 1 carbon is removed, forming carbon dioxide as a byproduct. Electrons are stripped, forming NADH. Coenzyme A attaches to the 2-carbon fragment, forming acetyl-CoA. Coenzyme A is removed, and the remaining 2-carbon skeleton is attached to an existing 4-carbon molecule that serves as the starting point for the citric acid cycle. The new 6-carbon chain is partially broken down, releasing carbon dioxide. Several electrons are captured by electron carriers, and more carbon dioxide is released. The carbon dioxide that you exhale comes from the reactions of cellular respiration. 2 ATPs are produced by the citric acid cycle for each molecule of glucose. At this point, only a small number of ATPs have been produced. However, more energy is available in the electrons that are being transported by electron carriers. While the citric acid cycle starts another round, let's follow an electron carrier to the next step in the process.
I hope you heard in that video clip then that in the Krebs cycle, all of these events are occurring inside the mitochondria, which means that we're going to find Krebs occurring in cells that possess mitochondria, which eliminates most of the prokaryotic cells. Well, it eliminates all the prokaryotic cells because they don't have mitochondria. So when we look at the events of Krebs, we are considering cells that have the double membrane-bound organelle, the mitochondria. In Krebs, the carbons that remain from glucose are given off as carbon dioxide, and then the important part in terms of energy transfer is the movement of the electrons onto NAD, FAD, and ADP as the energy is transferred. And so in that process, remember we had acetyl coenzyme A come in as the remnant of the glucose, at least half of it. It joined with a 4-carbon compound, and then went through a series of reactions to strip away the carbons, producing carbon dioxide, and then that carbon dioxide was given off as a waste product. The hydrogens from the remnant pyruvate are transferred over to NAD, forming NADH. Again, some carbon dioxide is given off as we lose the second carbon from this cycle. We have some ATP formed during the process of Krebs. And then also a FADH2 is going to carry electrons over to oxidative phosphorylation. I also wanted to point out that this happens twice for each glucose molecule that is oxidized. So a second coenzyme A is going to enter into the process. It will repeat the entire process, forming NADH a couple of times in the process, as well as some ATP. And we're going to have FAD reduced and then an additional NADH produced in this process. So one cycle of the Krebs cycle would oxidize the pyruvate. So it takes actually 2 turns of this cycle in order to completely oxidize or get rid of those carbons and produce all the carbon dioxide, so that we have all of the hydrogens moved over to a carrier and the carbons are given off as carbon dioxide and energy transferred also over to ADP, forming ATP in the process of the citric acid cycle. So that initial acetyl coenzyme A brought in the carbons along with those hydrogens and then joined with the 4-carbon compound, oxaloacetate, forming citrate or citric acid, which is sort of the keystone chemical that's produced, and oftentimes the name citric acid cycle is applied to these events because of that first formation there. Then a series of events occur producing NADH as well as carbon dioxide, removing 2 carbon dioxides. We also have ATP formed as well as FADH and then NADH. So through that process, we've pretty much dismantled the entire glucose molecule now. We need to think about, where did each atom in the glucose molecule end up when it was taken apart? Where did the carbons go? What did they produce? I hope you're thinking carbon dioxide. We had carbon dioxide formed during pyruvate oxidation, and then the remainder of the carbon dioxide was formed during the Krebs cycle. I don't want to call too much attention to carbon dioxide though, because remember, it's a waste product. It is not part of our desired energy end product. So really the part of your food molecules that are important are the hydrogens. Where did they go? They were transferred to the coenzymes, or the electron carriers, in this case FAD and NAD, and they are going to move those electrons along with that energy over to oxidative phosphorylation. So when you think about, you had to eat to get all these atoms into your body, remove the bulk of them, the carbons and the oxygens, really to get at hydrogens. It's just amazing to me that hydrogen plays such an important role. It's such a simple atom, yet our bodies can do some fairly amazing things with just those atoms. So that's what we need to know about Krebs. Those are the most important things. You need to be able to identify the energy-related end products, how the ATP was formed, what were the waste products, where did these events occur, what kinds of cells can go through Krebs, and where are these electrons headed. So you don't have to be able to trace every event or identify every enzyme involved in the process. These are the big picture questions you need to be able to answer. So I encourage you to stop the video for a moment, write the questions down, write a response to each of those, and be ready to discuss those questions and answers with your teacher. In general, you should be able to tell, what did we get from Krebs? What was the end result of all of that action that we saw occurring? And the primary events were that we had NADH produced, because that's going to be carrying electrons over to the electron transport chain in oxidative phosphorylation, as well as FADH2 produced, which similarly is headed to oxidative phosphorylation. We also had ATP formed by the substrate level phosphorylation. And then we have those carbon dioxides that are given off as waste products. So you may want to jot a few notes down on your diagram that you have in order to keep up with what is really important about what came out of and what goes into the citric acid cycle as we get ready to look at oxidative phosphorylation our next time. And so remember, we're trying to move that energy, harvesting that free energy. And so now we have pretty much taken apart or dismantled the glucose. And in our next steps, as we look at oxidative phosphorylation, we'll see what happens with all those hydrogens that got moved. So here's how we'll wrap up today's lesson. I'd like for you to watch the video that follows. It's fairly short and it summarizes the Krebs cycle. And then I want you to watch it again, but the second time, turn the sound off and see if you can narrate the events, paying particular attention to the important energy end products as well as any waste products that are produced in the Krebs cycle.
The oxidation of glucose continues in the Krebs cycle. Pyruvic acid molecules formed during glycolysis diffuse from the cytoplasmic fluid into the mitochondrion, but pyruvic acid itself does not enter the Krebs cycle. Pyruvic acid is oxidized while NAD+ is reduced to NADH, which stores electrons. A carbon atom is removed and released in carbon dioxide, leaving a 2-carbon fragment. Coenzyme A, or CoA, joins with the 2-carbon fragment, forming acetyl-CoA. One molecule of acetyl-CoA enters the Krebs cycle. The 2-carbon fragment of acetyl-CoA attaches to a 4-carbon molecule in the first reaction of the cycle. In a series of steps, bonds break and reform. 2 carbon atoms are released one at a time in molecules of carbon dioxide. Electrons are carried off by 3 molecules of NADH and one other molecule, FADH2. One step produces an ATP molecule. The 4-carbon molecule is regenerated. Since 2 acetyl-CoA molecules are produced for each glucose molecule broken down, a second acetyl-CoA enters the Krebs cycle. The same series of reactions occurs, releasing carbon dioxide and producing more NADH, FADH2, and ATP. The cell has gained 2 ATPs that can be used directly. However, most of the energy originally contained in the bonds of glucose is now primarily carried by the NADH molecules.
Our next topic in cellular respiration is going to be oxidative phosphorylation, where we finally get a chance to look at what those hydrogens are doing in the final stages of this process that are so energy efficient that allow us to produce such a large amount of ATP in this final step of oxidative phosphorylation.